From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on December 12th, 2025. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 119th episode of Matters Microbial. Thank you for every view or listen. I appreciate every listener and viewer. And please spread the good microbial word. My last microbiology class at the University of Puget Sound is nearly at an end, and the Bio 350 Micronauts have been quite wonderful to me, and it's a really great way to finish teaching. I like to teach Micronauts about the dangers of what I call swab stories, alarmist media stories about how bacteria are everywhere, often found by swabbing from a surface onto nutrient media, and in fact, microbes are everywhere, and most don't care about us at all. They're what I call meh microbes. They were here first, after all, so we shouldn't be surprised. First evolved, last extinct is a motto of my microbiology courses. One Micronaut found this swab story image from TikTok, as you can see, a post perennially popular about microbes on toilet seats, but also one about microbes on our unwashed pillowcases. I'll bet they're mostly skin bacteria and really are no big deal. Another Micronaut created this great meme about bacterial compartments seen here. Over and over again, we hear that only eukaryotes have organelles. Now, organelles are specialized compartments in cells that carry out specific functions. Do they never occur in bacteria and archaea, or do they only occur in eukaryotes? Well, the carboxysome, found in many autotrophic bacteria, is a compartment that fixes carbon dioxide into organic material using a popular enzyme, Rubisco. So, bacteria don't have organelles? The confusion comes from the fact that many bacterial compartments, often called microcompartments, use protein to bound them, but you can also find membrane-bound ones. So what I'm trying to say is when folks state that bacteria don't have organelles, I still see this eukaryocentric mistake in textbooks, to this day. I will continue to preach the one true faith of microbial centrism, never fear. My Micronauts also had a nice end to lab sessions with a final LuxArt exercise. As before, the students paint with luminous bacteria onto nutrient media, incubate overnight, and the next morning photograph their work. It's a glowing experience for all. I had the Micronauts first work with standard size Petri plates and write their names, and as you can see, Micronaut Shiana was quite creative. Micronaut Ava, seen here, showed a certain degree of self-confidence. The larger plates had enough real estate, that is to say space, for some wonderful LuxArt. I'm going to create a video showcasing their work, but here are some examples from this great class from last week. Micronaut Jonah painted one of our local mountains, seen here. Micronaut Layla was clearly influenced by the television series Stranger Things, with a glowing demogorgon. Micronaut Ruby, who often had fungal contamination on her plates, did a nice job of showing fungi spreading spores. Micronaut Hannah was impressed by the isopods that I raise to feed Azula, my African fire skink. And finally, Micronaut Shannon created a penguin with a petri dish and inoculating loop. I truly enjoy putting on these LuxArt workshops and will be at Penn State in April doing one. I hope to continue to get invitations. It's a wonderful and fun way to promote microbial literacy in the intersection of science and art. One of the things that amazes my microbiology, filled with what the microbe and overwhelming microbial greatness, is that even the best known bacteria and archaea have genomes with 20 to 40% genes of unknown function. Researchers sometimes call these unknown genes microbial dark matter. I really love that term. So I'm very pleased to introduce Dr. Julia Willett of the University of Minnesota to the podcast to discuss the work that she and her colleagues do as they investigate microbial dark matter in microbial genomes. Welcome to the Quality Quorum for today, Julia. It's great to meet you.
Hi, Mark. It's so nice to be here. Thank you so much for having me.
Well, I think a good place to start is talk a little bit about what you mean by microbial dark matter.
Sure. So I love to use the term microbial dark matter to, refer to these genes that we haven't really shed light on yet, if you will. I think we are maybe we learn in class, or we kind of have this assumption that if you take E. coli, for example, which is used ubiquitously in research labs, both as a tool for genetics and for things like passaging plasmids, there's this assumption that we know everything that E. coli does. We know everything about it. We know what every gene does. And that's really not true. And so I love this idea of dark matter, or this analogy of dark matter, to kind of convey the fact that bacterial genomes are this really kind of mysterious place that we really have a lot to uncover.
It's interesting that you would say this because when I teach about this stuff myself, and we'll say, here's the genome size of E. coli, and then here's an estimate of the number of genes. And we have an ability in many organisms like E. coli of individually knocking out each open reading frame that we suspect of being a gene, and then seeing what the effect is. And as you say, the really, I don't know to call it frustrating or fascinating, maybe both, is that there are some genes, they're clearly expressed, they clearly are there, but they're related to nothing we know of, and yet if you knock them out, it's non-viable cell. So they're doing something important. You and I, even before this started, talked a little bit about the mycoplasma that has this completely defined genome. And there's still a decent percentage of unknown genes in that defined organism that are necessary for life.
Yeah, and that's a fact that I love, and I think it just really highlights this kind of issue of dark matter or unknown genes, that if you take basically the smallest viable genome, or what we think is the smallest viable genome, it's really amazing that we don't know what sort of 20% of the basic function that a cell needs to survive. We don't know what those functions are. An analogy that I really love to use when thinking about microbial dark matter is that of an iceberg. So if we think about an iceberg in the ocean, we can see the sort of part that's sticking out above the water, but that's of course usually a very, very small percent, or a very small percent of the mass of the iceberg, because so much of that is hidden under the surface. And so I think this is a great analogy for microbial dark matter, because really, the number of genes where we can be really super confident that we know what they do in any given organism is kind of that small part of the iceberg that's sticking out on top of the water, and the rest of gene function is kind of that mass that's hidden beneath the surface.
And for listeners and viewers, what can often be done is you'll get what's called an open reading frame, where there's a promoter-like sequence, there's a terminator-like sequence, so you know that it's being transcribed into RNA, and you take that open reading frame and you find out what other sequences where we know the function of them are related to it. And so when Julia and I are talking about things that are unknown, all of these programs that do annotation, as it's called, come up empty. It's happened in my own lab, and you have no idea what it does. And this is something that is so antithetical to how I was trained. It was very reductionist, and we were looking at individual genes, but that's not how life is. And so what's deeply exciting about the technology's improvement is the ability to, as you say, look at genes in context, what I call the genography of what's going on.
Yes, absolutely. And, I think thinking about the context of an organism is so like absolutely essential when we're thinking about discovering new functions for these uncharacterized bacterial genes. And so, one of my favorite examples of this is in the organism that my lab primarily studies, which is Enterococcus faecalis. E. faecalis is not motile. It doesn't sporulate, but if you look in the genome, you find, half a dozen to a dozen genes with annotations or predicted functions that are related to sporulation and motility. And so when we find that these genes are differentially expressed, in an RNA-seq experiment, or if we find a phenotype for them in a genetic screen, this adds kind of an extra layer of a challenge to figuring out what the actual function of these gene products are, because we know that those sort of annotations aren't relevant to what we know about enterococcal biology. So, another example that I really love is what the Pseudomonas community has done to really advance genome annotation, especially in Pseudomonas aeruginosa. So, the Pseudomonas community has built this really beautiful website and this really beautiful database where they've kind of painstakingly over the years gone through and really carefully curated the annotations of many Pseudomonas aeruginosa strains. And they've come up with this classification scheme where they have essentially four classes that they use to define what is known about gene function. The first class is kind of the most stringent, and this says that the function of a gene or a gene product has been experimentally demonstrated in Pseudomonas aeruginosa itself. And as we were just saying, that's a great bar because you're saying you know what the function is, and you know that that's important within the context of the organism that you're studying. And then from there, you can say, okay, well, maybe in the second sort of subset of what's known about gene function, the function hasn't been shown in Pseudomonas aeruginosa, but it's been shown in something similar. You can move down another level and say, maybe this is something that's based on annotation, for example. And then sort of the final layer is, there really isn't any similarity to anything else that's out there, or it's only similar to other genes of unknown function. And I think this is just such a beautiful example of kind of the layers there are to really understanding gene function, not only from a kind of computational prediction level, but from actually showing experimental evidence in an organism.
I think this is such a good system to look at, and the Pseudomonas community ranges from folks who work on medical relevance to environmental, soil composition. And this brings us into this concept which my students dislike hearing about pangenomes and that kind of
Mhm.
thing, but it's the way it really works, and having a large group of people working together.
Mhm.
And I keep thinking when I say Pseudomonas community, it's kind of a pun, which it kind of is, but it's also true that you need to have context and people that you can you can work with on that.
Yes.
And that must have been very fine to do.
Mhm.
And I wanted to say parenthetically a couple of examples of what you mentioned. So the bacterium that lives within the giant tube worms
Mhm.
at hydrothermal vents,
Mhm.
Riftia. And not much is known about how those organisms run around. And when people were able to pull genomes out of the bacteria within Riftia, they were able to show that there were motility genes, and those motility genes worked in E. coli to correct for different loss of function mutants.
Mhm.
So that gave them some insights into perhaps how those symbionts are moving from place to place.
Mhm.
But I think the thing that you would appreciate the most, and I'm sorry for the old man discussion here, is that many years ago I was asked to review a paper on the genome of Delavibrio, one of my favorite
Mhm.
organisms. And it was interesting, these very famous people who I have such respect for were writing about this.
Mhm.
And what they said at one point in their paper is that many people thought that Delavibrio was stealing ATP from their prey cells
Mhm.
as a way of saving energy.
Mhm.
But in fact, there were no genes relating to the ATP transporters known in Chlamydia and Rickettsia, which are both bacteria that live inside eukaryotic cells and steal ATP from those cells.
Mhm.
And I looked at that and I thought, well, hold on. And when you look in PubMed or Google Scholar, you discover there are multiple papers biochemically showing two different mechanisms by which ATP is translocated, transported in Vibrio. And what that means is that there were transporter genes, they just weren't related to the ones that were known.
Right. Yes, and that's such a great example of I think this kind of issue of dark matter. Of I think bacteria are smart, right? That bacteria have been doing this a lot longer than we have, and they have a lot of ways of doing what they need to do to acquire nutrients and to survive under stressful conditions and to adapt to the environment that they're in. And so I think that, some of these dark matter genes, right, might be another way of doing something that, the cell really needs to do in order to survive. And that kind of brings us to this big question of, is studying single isolates of very well characterized strains in a test tube really the best way to discover new gene function? And I think that we really just have so much to uncover, and all of these new tools that are being developed that allow us to culture things that were previously, we were unable to culture, or to grow things in different ways, I think is really necessary to kind of uncovering these layers of unknown gene function.
Many times in this podcast series, folks have talked about horizontal gene transfer, where, for example, a gene encoding antimicrobial resistance can be transferred from cell to cell. Is there any evidence of this uncharacterized genes, this microbial dark matter, also being subject to horizontal gene transfer and maybe revealing more about their possible functions?
That's a great question and a really interesting idea. And if you look at plasmids, for example, which are, things that are very commonly horizontally transferred from one organism to another, there are definitely uncharacterized genes or these dark matter genes that can be found, especially if you start looking at these large, conjugative plasmids that are maybe, 40, 50, 60 kb, that there are definitely dark matter genes or uncharacterized genes that are getting kind of shuttled around from organism to organism. And that's a really fascinating idea about whether we can maybe leverage different organisms, different strains, different genetic backgrounds to learn more about their function.
It's interesting that you'd say this because my own background, we originally assumed that a lot of genes in organism A would not work in organism B, but sometimes they do. And so I've taken that away with me. And so sometimes I often worry about that. I think about that the idea that perhaps a gene that isn't functional or doesn't do something clear cut in one organism might make more sense in another.
Yeah, absolutely. And, especially if you think about redundancy, or if you have something that is an uncharacterized gene and it's in an organism where there's already a gene or a gene product that is doing that exact same function, if that gets transferred into a different organism, that might be a better background for studying that function.
Oh, it's so interesting like that. How did you become interested in this topic? Was there an important point where you kind of, as you see, it's much on my mind what we don't know? Is there some light that, because so many people will study a particular model system and they'll fall in love with it, and I appreciate that. But I always have been fascinated by the things we don't know.
Yes. Yeah, that's a great question. And there was really kind of a light bulb moment for me, and really getting interested in this idea of unknown gene function and microbial dark matter. So when I was a graduate student at UC Santa Barbara, I worked on these contact dependent growth inhibition systems that are widespread in gram-negative bacteria. They're a really beautiful system to study. And one of the projects I worked on during my PhD thesis was trying to understand how toxins that were made in one cell could be delivered into another cell, right? That's a big challenge. In gram-negative bacteria, you have to go from outside the cell across two membranes if you're trying to reach an intracellular target like DNA or RNA, and that's a really complicated process. And so I was doing a lot of genetic screens, and the sort of first gene that I hit, where the transposon insertion, conferred resistance to this toxin system I was studying, was in a gene called YCIB. And of course, in E. coli, the subset of genes that all start with the letter Y were annotated that way because at the time the function was completely unknown, and so they're what's called the Y-ome of E. coli, this sort of subset of uncharacterized genes. And so this was super exciting. I'd never heard of the Y-ome before. I'd never really heard of, an uncharacterized gene before. But it was also a real challenge because, when you're studying the activity of something that's functioning as a receptor, you would like to be able to, do some mutational analysis and try to break the function of maybe transport or whatever, the normal function of that receptor is, and see if that's required for the toxin system that we were studying. But because this was an uncharacterized gene, that was a real challenge. And so I think that was really kind of the first light bulb moment for me where I went, wow, we really don't know a lot about bacterial genomes.
Who did you work for at UC Santa Barbara?
I worked for Chris Hayes.
You may or may not know that I've met you.
Yes, at the Asilomar meetings. Yep. Yeah.
I've been to your talks. Yeah.
Oh my gosh. Such a small world, really.
It sounds like a pun that it's a small world in microbiology, and I guess it is. But this is what I tell my students. It's very important to be polite because you will see other people again.
Mm-hmm, mm-hmm. Yes, very much.
Yeah, but
so.
as I'm listening to you and you're talking about the Y-ome, I go, that the penny dropped.
Yep. Yeah. Yes. So that moment in grad school was really kind of, the first thing that set off my interest in these uncharacterized genes. And then, when I was doing my postdoc research, I trained in the lab of Gary Dunney at the University of Minnesota. And Gary's lab had really undertaken this kind of enormous effort to build transposon libraries in Enterococcus faecalis. And this has been such a valuable resource, for the field, and it's really just kind of robust platform that allows for really kind of robust genetic screens in E. faecalis. And Gary's lab had created this sort of subset of the transposon library where the only mutants that were included were in uncharacterized genes and then in intergenic regions, because as you alluded to earlier, right, we don't just have genes in our genome, or bacteria don't just have, nice genes in their genome. They encode all sorts of regulatory RNAs and all of these molecules that maybe are made from intergenic regions that, again, are really fundamentally important for the things that bacteria do. And so I had done a genetic screen using this sort of uncharacterized gene intergenic region transposon library. And I got a lot of hits that were related to biofilm formation. So I was essentially looking for genes, specifically genes of unknown function that were required for E. faecalis to be able to build a biofilm.
So a couple of things for listeners and viewers. Why should people care about Enterococcus faecalis? Now, you never have to justify this to me, just in general, but people love to hear that.
Yes. Yes, that's a great question. So Enterococcus faecalis is a really fascinating bacterium. So it is a gram-positive pathobiont. So it resides in the gastrointestinal tract of about 80% of healthy adults. And it doesn't really cause any issues there, but it can become very problematic after things like long-term antibiotic treatment or in patients who are immunocompromised. It can overgrow or bloom in the GI tract, and then it can disseminate out, to other body sites. And outside of the GI tract, E. faecalis causes a variety of infections, everything from bloodstream infections to wound infections to prosthetic joint infections. And a lot of these infections tend to be biofilm associated. And so the bacteria themselves are sticking to things like the surface of an implanted, prosthetic device, for example. And biofilms, of course, are very hard to eradicate. They can increase antibiotic resistance, and E. faecalis can be pretty intrinsically resistant to antibiotics as well. And so it has this really interesting dual life cycle where it can survive in the GI tract, which is this very harsh environment, really without causing many problems. But under the right circumstances, it can kind of transition to this very problematic sort of infection state. And it's a really interesting organism to study for those reasons.
I'm really happy to hear you say this because Arturo Casadevall does not like the term pathogen.
Hmm. Hmm.
And it's so funny. I used to have an assignment with students where they would write about it, and one of them did like a haiku, and they made a mistake, and they said pathogon instead of pathogen.
Ah,
And
interesting.
Arturo loved that because he wants that particular phrase removed. And the
Mhm.
reason that he says that is it's all about context. You know, like
Mhm.
as a real estate agent says, location, location, location.
Yes.
And so one of the problems that we fight about in terms of microbial literacy is the idea that microbes are everywhere, even things that you could potentially have a disease from.
Hmm.
They're just not in high numbers. I mean, I know for a fact, because I've done the experiment, if you swab skin on people and use PCR to look for some of the toxin genes that you see in MRSA, yeah,
Hmm.
you'll see them. They just haven't gotten a foothold. It's
Right.
context. You know,
Yes.
it's the right neighborhood, right?
Yes, absolutely. And I think Enterococcus faecalis is, yeah, such a great example of, like you said, location, location, location. And that's one other thing I really enjoy studying about it, because what it allows us to do is kind of ask related research questions, but in different
Yeah.
sites in the body. And so, for example, we can ask what uncharacterized genes are required for survival in the GI tract, and we can compare that to what uncharacterized genes are required for survival in the wound, or in a simulated wound condition or something like that. And so it allows for this kind of comparison across different body sites, and again, kind of more insight into the functions of this microbial dark matter.
So I also wanted to get kind of take a moment and have you explain a little bit. Many people don't appreciate what a transposon mutant is.
Mhm.
I've been working on them for many, many, many years. And I think that's a good thing to tell listeners and viewers about, and more specifically about the library that you have,
Yes.
because that's something that when I was first doing transposon mutagenesis, I only dreamed of.
Yes, it is. It's a very special resource. So we do a lot of work with transposon mutants. And so what each transposon mutant is there's basically a piece of DNA that gets inserted into the genome of the strain that you're working with. Our transposon system uses a Mariner transposon, and so we get insertions at TA dinucleotides in the genome. The genome, has many, many TA dinucleotide spots, and so if we take a very large collection, say, 100,000 colonies, for example, there could be up to 100,000 different mutations or different transposon insertion mutants that are kind of represented in that collection. And when you're thinking about transposon mutants and using them for genetic screens, there's sort of two ways that this can be applied, or sort of two ways that you can use these libraries. The first is, I think the way maybe a lot of us were trained to make these, or the way a lot of us do it, which is where you kind of gather up all of your colonies and you combine all of them and you grow them in a certain condition, or you challenge them with an antibiotic, and you use this pooled approach. So all of your colonies are pooled together. And that is a great approach for so many reasons. But then if you get your sequencing results back, or you find a gene that you're really interested in, a lot of times what you need to do is then go back to the strain that you're working in and make a mutation de
Yeah.
novo. You need to knock out the gene. You need to try to, disrupt this gene so you can start studying its function. And what Gary's lab really beautifully did is they built what's called an arrayed transposon library. And so they created this large pool, or this large set of transposon mutants, and then, very painstakingly, one by one, went through and pulled individual colonies and arrayed them in 96 well plates. So we have a collection of 73 96 well plates, and every well has, one single mutant in it, or one single transposon mutant in it. And so when we do these large genetic screens, we can maybe use a pooled version and then immediately go back and get the mutant that we're interested in.
And again, for listeners and viewers, when these pieces of DNA, and please forgive me putting it this way, and it hops into a particular target gene, because of the physical insertion of DNA, it reduces or eliminates the expression of that gene.
Mhm.
And it was very powerful when it was first discovered all these years ago and used by so many important people in microbial genetics. But what's interesting here is that you now have, and there are other systems like this as well, where, as you say, you have in your -80 freezer a rack of these of these 96 well plates
Mhm.
where there's an insertion into essentially every gene in that organism.
Mhm.
And as you say, for the pooling technique, you can narrow things down and then get to that one insertion,
Mhm.
which is just beautiful.
Yes, they're such a fantastic resource, and I'm so grateful that this library exists and that we can use it. But I think it's also just such a powerful tool for the field. And my lab now sort of has taken over curation and distribution of this resource, and we're always very happy to sort of, if anyone needs the full library or just a handful of mutants, we are really kind of dedicated to making sure that this resource is being shared really as widely as possible, because it does really facilitate a lot of work and a lot of genetic screens. And like you alluded to, these arrayed libraries are not unique to just E. faecalis. There's arrayed libraries that exist for a number of organisms now, Pseudomonas, I think, being one of them. Yes, and they're fantastic. They're just really fantastic genetic resources to have.
I mean, such an incredible amount of work, number one,
Hmm.
to put them together. But
Hmm.
then number two, and I want to emphasize this because sometimes scientists are not portrayed. Well, okay, it's because of what people see in the media,
Hmm.
right? And I'm not being political here. I'm even talking about movies and television shows.
Right.
The fact of the matter is, most scientists that you meet actually do share a lot of what they find with other people.
Hmm.
And this is a wonderful example of it, because if everyone who studied Enterococcus had to recreate that matrix, I can't begin to tell you how long that would take.
No, I did the time, the effort, the costs that it, yeah, that's why it is so important to me and is really, kind of one of my lab's fundamental interests is, making this widely available, is because, like you said, if everybody had to recreate the same tool, I mean, it's just reinventing the wheel. Truly, it would just be trying to do the same thing over and over again. And yeah, I think it's really incredibly important that not just our library, but so many of the resources that are available through the E. coli stock center and the bacillus stock center, for example. There's a Staph aureus MRSA transposon library that's really
Right.
widely available through the University of Nebraska. These are just all phenomenal resources that should be shared widely.
And it's so funny because there are so many organisms that I'm interested in where there aren't good tools available.
Hmm.
And that's the balance of things,
Yeah.
because I was always interested in, this is a big surprise to anyone who knows me, the weird ones. Okay. That's just me. Always been that way. Always will. That's just how it is. But you have, in many cases, an inability to do things,
Hmm.
tools that aren't yet available. I mean, I had to develop some of my own in graduate school for that reason. And there's a place for that. But if you're working in a system where there's a community, such an advantage on so many levels, not just material, but people who are willing to take their time chatting with you about it.
Yes, very much so. And I'm very lucky that the Enterococcus community is very strong and very supportive. So it's a fantastic group. Um,
So what were you going to say? I'm so sorry.
No, I'm so sorry. I was going to say, to your point, and kind of considering this microbial dark matter, a lot of the quote unquote weird organisms, or the ones where we don't have these transposon libraries, I think that is where we really have so much to discover about basic bacterial gene function, that if no one has ever looked, no one's ever studied the function of these genes in this particular context. Yeah, I think with the advancement of new genetic tools and the ability to culture things we've never been able to culture before, or do genetic manipulation, that will really contribute to our efforts to study uncharacterized genes.
During the last session, I put up a quote by someone who's a colleague of mine for many years, Bill Metcalf at the University of Illinois, Urbana-Champaign. And he was making fun of something that I was doing, where I was using a phenotypic screen, and I was very excited by it. And he told me it, And I made it look more formal last time. He goes, Mark, those phenotypic screens are not for the bacterium's benefit. And the point I'm trying to get at with this is that when we try to study an organism, we have to do it with our human senses and the human ways of thinking when they have their own way of doing things. And I don't want to sound vitalist with you, but I don't think we have a microbial perspective. And that's why I'm so excited about the approach that you talked about, which I believe we can think of almost like a subtraction event where you take this library of Enterococcus mutants and you pass them through a selection situation to find out which ones don't grow in the gut normally, which ones don't appear in wounds.
Right.
And then you hope to find that those mutants are involved with the process of surviving in the gut or proliferating. And that's exactly how you found some of these biofilm forming genes. Am I right?
Yes, that's correct. Right. The screen that I did when I was a postdoc was to take this transposon library with all of these uncharacterized gene mutants and to do a very simple screen of growing it in a 96-well plate. Enterococcus, especially Ephecalis, loves to stick to surfaces, almost all surfaces except for glass, which I think is a very curious sort of fact. Yeah. But I grew them in 96-well plates. And then, I came in with a dye and I stained for the biofilm biomass that was there. And you get this really beautiful colorimetric visual readout of wells that have a lot of biofilm stained bright pink because of the dye we're using, and wells where there are not a lot of cells that are attached or there's not much biofilm are more clear. And so I could quantify that data and make this kind of distribution. And then I could select the mutants that were unable to form a biofilm and go back and retrieve them from this transposon library. But you're right. Really what I was doing was asking the cells to do what they would be doing anyways, which is to grow in a 96-well plate and attach to a surface. And then I came along and just measured whether or not they could do that.
I think it's just a fascinating approach. And again, I'm used to the idea of looking for a defect and seeing how it applies in other contexts. But what's good about this is you're genuinely using perhaps not in this exact experiment, but in related ones that I know you've done, where you're letting the organism show you what's needed.
Right. Yeah, absolutely. And I think the other kind of powerful thing about this approach is, we start with a phenotype. I am of the opinion that you need a phenotype to study anything. And so a lot of experiments in my lab start by trying to find a phenotype. But then once you've found that phenotype, the challenge with the uncharacterized genes is that we can't just go to KEGG or, EcoPsych or your favorite database and look up what they do, right? And so I think the other, advancement in the last several years that I'm really excited about, kind of revolutionizing our quest for discovering microbial dark matter, is AlphaFold and really these, advanced computational ways of predicting the structure and sometimes the function of proteins, because what we've been able to do is kind of couple these. So we can take a phenotype and we can couple this with computational predictions of structure and function. And we can then use that to kind of prioritize or narrow down a very large list of uncharacterized genes into things where we can start to form testable hypotheses about, their actual function.
That is really cool.
And I think I have a schematic kind of showing an example of that. And so one thing that came out of the screen that I did when I was a postdoc was we did this big biofilm screen. I did a lot of structure predictions. This was using Fire2 back in the day because AlphaFold wasn't out yet. And what I was able to discover was a previously uncharacterized phosphatase in Ephecalis. I was able to then go and purify the enzyme, show that it had phosphatase activity in vitro. We were able to name this gene, so we named it BPH for biofilm phosphatase. And so now, a future project in the lab or something I'd be really excited to pursue would be trying to understand, okay, we know this is a phosphatase, we know this is important for biofilm formation on kind of an overall scale, but what about this phosphatase activity inside the cell is specifically important for biofilm formation?
That's amazing stuff. And something that, and I get over, okay, I get enthusiastic. Overly enthusiastic was just the definition of enthusiastic with me. But I mean, there's so many wonders to observe and to see.
Mhm.
And to me, it has always been so exciting. I was excited when you could only look at one gene at a time. So
Mhm.
just imagine how I feel today.
Yes. Absolutely. I think the ability to do these really large genome scale screens, especially if we're talking about something like transposon sequencing, using these pooled transposon libraries that every year, new sequencers are being developed and the cost of sequencing is coming down. And there's all these advancements, like barcoding with the development of transposon libraries, that it's a really exciting time to be a bacterial geneticist.
Oh, yeah, I couldn't agree more.
Yeah.
That's why I feel badly about entropy having its way with me, because the kinds of things that I would love to do are what you all are doing now.
Mhm. Well, we're very grateful that you have trained some very amazing people who are going on and having their own fantastic independent careers and, carrying your love of bacterial genetics throughout their own research careers.
Thank you so much for saying so. It's something very gratifying. So I really like this idea of you trying to make predictions. I thought your phosphatase example was especially relevant.
Mhm.
What other things have you found out that you would care to share with us?
Sure. So we have found that maybe surprising, maybe not surprising, number of uncharacterized genes in Enterococcus are really important for modifying the cell surface. And this makes sense if we think back to, location, 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 that the outside of the cell is what's in contact or, in contact with the environment around it. And so it makes sense that, we all know bacteria need to adapt and change to the environment that they're grown in. And again, kind of thinking of this idea of if you grow something in a test tube, you find out what's important in a test tube. But if you
Yes,
grow
you do.
it in a different environment, you find out what's important in that different context. And so I'm really excited about a lot of projects that are still kind of in the early stages, but seem to kind of be coalescing around this idea that there's a lot of uncharacterized genes that are involved in that sort of first interaction that E. faecalis might be having with its environment at the cell surface.
Are you going to share anything, or is it too early?
It's still too early. We have a lot of like unpublished data, and it's all in that stage where it's like I would love to talk about it, but I don't know how long it's going to take me to get this manuscript off my desk.
Oh, of course, of course.
So,
But again, it's such an interesting thing. So, let me throw something at you.
mhm,
In some of the work that I do, when I'm studying luminescent bacteria, and I am
mhm,
finding things that predominate as DIMs that have nothing to do with the actual lux operon that
mhm.
I'm interested in, but I get these unusual other genes, and sometimes I get uncharacterized from the annotation software. What would you recommend I do next?
Yeah, that's a great question. So, whenever someone comes across their favorite uncharacterized gene, my which I think maybe we all have our favorite uncharacterized ones.
That
My
that's
in,
a that's a t-shirt, don't you think?
I do think so. I would wear that.
What's your favorite what's your favorite uncharacterized gene? I love that.
I think that would be a hit at a conference. I think you'd get a lot of feedback.
Yeah.
Um, but whenever someone comes across an uncharacterized gene and they're like, I'm really trying to figure this out, my first advice is always to turn to sort of computational predictions. That I mentioned AlphaFold earlier, but again, seemingly every week or certainly every month, I'll see a new preprint about a new database or a new annotation pipeline or something that's really designed to kind of get at this question of how do we best use computational resources to make insights into gene product or protein function. And so my advice is always, go to as many databases as you can. Don't just rely on AlphaFold or, a sort of one prediction. That try to find everything that you can about any domains this might have, what other organisms it's conserved in. Are there any kind of conserved motifs that you can use to maybe try to find an active site of an enzyme that would allow you to do something like alanine scanning? But really just kind of go to the computer and go far and wide and really just learn everything you can at the computational level about this gene or this gene product.
And this is the kind of thing that I really do tell students is that I'm not saying that microbiology has become computational.
Hmm.
It's like, to me, like saying something like microbiology is all about agarose gel electrophoresis.
Hmm.
You're certainly going to use that. It's
Hmm.
part of what you do.
Hmm.
And now here's another set of tools that you need to have, and that's going to be the use of these databases.
Yes, absolutely. That I really do think that it has to be sort of a two-pronged approach, right? You need the sort of classic genetic screens, you need the phenotypes, you need those sort of experimental techniques, and then you can couple that with the computational predictions and with these annotations. But I think that, one or the other is probably not going to get necessarily the answer that you're looking for. And yes, I agree. I don't think that we've moved completely to a computational microbial world at all.
Oh, and people accuse me, and I understand why they do, of hating on structural genomics.
Sure.
And I don't, but I found this to be a lovely thing that you said, and it's something that I've said for many, many years as well, that the phenotype is what interests me, the
Mhm.
change in what you observe. So there is nothing wrong with just looking at DNA, especially
Mhm.
if it's an organism you can't cultivate, because
Mhm.
what choice do you have,
Yes.
right? But I will say, I would look at these kinds of things and say, okay, so now I have a prediction. If I knock out this unknown gene, I have a prediction what the effect will be. Do I see that? And that's a powerful thing that we now have the tools to be able to do, even in undergraduate labs like my own.
Yes, absolutely. And I'm so glad that you brought up, really the power of getting students involved in research, especially at the undergraduate level. That really one thing I love about running my own lab now is the ability to work with so many amazing undergrads and to, I find it very gratifying when you can teach them how to do a biofilm assay and what the controls are, and then watch when they sort of stain and image their first plate and go, oh, okay, there's biofilm here and there's no biofilm here. I think that's a really fantastic part of my job.
We had an episode last week with Matthew Fields at
Mhm.
the Center for Biofilm Engineering.
Mhm.
And I had done some biofilm work with Pseudomonas putida many
Mhm.
years ago as something for first year students using crystal violet dyes and 24 well plates. And I showed some of those images last week. And you're quite right. When they are actually able to draw that distinction and see there's a dose dependent effect of something they're studying,
Mhm.
there's no substitute for it.
Yeah.
And I should say that what really works with students is not a demonstration of something, although you have to teach that way perhaps. It's when they come up with an idea and then they explore it.
Mhm.
That's the key part, because then they feel a sense of ownership
Mhm.
and a very natural enthusiasm that in all seriousness, it's kind of hard to get going when it's a demonstration that's been done year after year.
Mhm. Mhm.
And so when we talk about unknown microbial dark matter, unknown genes, you can just imagine what a wonderful thing that would be for students.
Yes, absolutely. I teach an undergraduate microbiology lab class now, and I have lots of ideas for how we can implement genetic screens and whatnot that I'm kind of working on building. But the way that the class is set up now is they're not doing uncharacterized gene function per se, but what they're doing is isolating and enriching specific organisms from soil samples and from water and whatnot.
Mhm.
They isolate bioluminescent organisms from fish, which is always a very fun activity. But I think the same purposes kind of apply, that you really see students kind of take ownership of these isolations. And we do some 16S sequencing at the end. And it's always great to then, be reading through the student lab reports and they say, well, I couldn't quite make a species identification, or I'm not really sure that what I have is well represented in the blast databases. And yeah, just that idea that it's something that they're doing and they're taking ownership of, and they're really thinking deeply about what this organism could be, and it's a great experience.
I think in many ways, education gets short shrift because of issues in society and government and all kinds of things that I'm not competent to comment upon, though I do have strong opinions. But I will say that I'm a big believer in letting students develop their interests and
Mhm.
supporting them in doing so.
Yes.
because they end up learning so much
Mhm.
and so much more than even they thought. I want
Mhm.
to emphasize that.
Yes, absolutely. And even, I have not been running my own lab for very long. And even in the few short years that I've been doing it, it's really just remarkable that we have amazing undergrads in Minnesota. There's amazing undergrads at every institution. And yeah, I've been so lucky to have just some phenomenal undergrads come through that bring their own ideas and bring their own perspectives and, bring something that they heard in class or they were assigned to read about, for a journal club, and bring that idea back to lab and ask how it applies to our biofilms and how it applies to our organism.
I couldn't agree more with that. What kinds of future areas do you think you're going to get into the lab?
That's a great question. I think right, I think it's a good problem to have. I think, a lot more ideas than time, or a lot more ideas than people. But like I alluded to earlier, one thing I love about studying E. faecalis is that we really can ask sort of core questions. And apply that to many different body sites. And so we've really been laying the foundation for this for the past couple of years. And I think this work will expand of asking what genes are required to build a biofilm, what uncharacterized genes are required for survival in all of these different niches that E. faecalis can kind of inhabit in the human body. The other thing I'm really excited about is I've talked a little bit about kind of the importance of the computational side of things with uncharacterized gene function. And there's a technician in the lab, Alex Snell, who's been working really hard on implementing some of these computational ideas to allow us to look at uncharacterized gene function on a really wide or genome wide scale. So I'm really excited to see how this work kind of continues to come together in the future.
I have to tell you, every time I do this podcast and talk to people like you, I sit here and there must be a German word for this where you're jealous and yet happy for someone. And so I look at this and think, boy, I wish I wasn't, I was not much use as an undergraduate, but I wish I was an undergraduate and have these kinds of opportunities, because I think it's a wonderful thing.
Mhm.
And I wanted to say, because this is the microbial elephant in the room, is that you have a former undergraduate student of mine currently in your lab who's a former podcast guest,
Yeah.
Ruth Eisenberg, and I am so delighted to find that connection.
I am as well. I am so lucky to be able to work with Ruth. And again, we kind of spoke earlier about what a small world it is, that it really is. And it's such a great example of, how much mentorship matters and how these connections and these skills that students build as they progress through the different stages of their life really contribute to their careers. That, yes, I'm very lucky to be able to work with Ruth every day.
I am a Ruth fan.
Same. I'm a big Ruth fan.
Did you know that we used to call her Ruthless, and I hope that
Uh-huh.
she continues to use that as time goes on.
Yes, I believe that is still her social media handle, so I think she's kept the nickname
Yes.
going.
Yes. So I wanted to say, as we're starting to close out on time a little bit,
Mhm.
what I was going to ask are two basic questions for you. And the first is there anything special about, we talked about the light bulb that went off in your head about
Mhm.
this research
Mhm.
light bulb, the bioluminescent bacteria in your head that went off about this. But I wanted to know what got you going in microbiology, because a lot of people I talk to think there's only one path to being a microbiologist.
Mhm. Yes, that's a great question, and I think my answer kind of comes back to the importance of undergrad research, like we were just talking about. So when I started college, I was a nutrition major. I don't think I really knew what I wanted to do with my life.
Mhm.
I think the only thing I knew is that I probably didn't want to go to med school, and I don't even think I had a good reason for that. It just wasn't something I was interested in. And so I decided to major in nutrition. And I think during my sophomore year, there was a new assistant professor in the Department of Food Science and Human Nutrition, Dr. Michael Miller. This was at the University of Illinois, where I went to undergrad. And Dr. Miller came and gave a guest lecture in one of my nutrition classes about the gut microbiome. And I maybe this was the time of Activia commercials that, the gut microbiome was kind of being talked about, but yeah, it was truly just one of those moments for me that it was just the most fascinating discussion I'd ever heard. And with all the confidence that I didn't know I had, I went up to him after class and said, this was so exciting. Can I do research in your lab? And he was very kind and very patient, and he said yes. And I just really valued that undergrad research experience because it was really the first time I'd worked in a lab kind of outside the traditional, lab class where you come in and you do an exercise. And one thing I really valued about that from sort of the microbiology perspective was that I think it was a very different microbiology than maybe I'd been learning about in my classes. That it was really a food microbiology lab. He worked on lactobacillus. And so I learned so much about kind of applied food microbiology and sort of all of these different angles of microbiology that I'd really never heard of before. So that was, I think, the first of two really kind of core events that set me down the path of microbiology. So the second light bulb moment that I think really set me on this path in microbiology was, again, when I was an undergraduate, I took bacterial pathogenesis with Dr. Brenda Wilson. And I remember that really was the first time that I felt like it really just clicked for me in a class. And I remember really vividly being in the engineering library and studying for an exam, and just thinking that it was fun, that we were learning about the type 3 secretion system and gentamicin protection assays and all of this, really fundamental microbiology work. And I was just captivated. And I had, kind of a winding path to get there through changing majors and changing labs and again, really not knowing kind of what I wanted to do after I graduated. But I think my first research experience and certainly that class really solidified my love of bacteria.
I'm so glad to hear this because so often I will hear from students that there's only one path. They think you had to be a cell and molecular major. You had to start research as a first-year student. Mm-hmm. Fill in the blank. I've heard it all. And the fact is, just as the microbial world is diverse, so are the paths that lead to the microbial sciences.
Yes, absolutely.
And, yeah. And you're just another example of that. And look at the enthusiasm that you feel for it, which certainly comes across. So the other thing that I like to do as we close out, if it's all right with you, is perhaps 3 takeaway lessons from today's session about microbial dark matter.
Yes, absolutely. So I think the first takeaway from our discussion of microbial dark matter is that we really know shockingly little about bacterial genomes, that whether it's, the minimal design genome, whether it's E. coli DH5 alpha that we're all, you make comp cells out of, or whether it's kind of your favorite organism. I think if you really look under the hood at the genome, we kind of know surprisingly little about what's there. I think my second takeaway is really that the organism matters, that I know, there's the famous saying of what's true for E. coli is true for an elephant. And I've heard a take on that's, what's true for E. coli is true for an elephant, but not bacillus. And I think that's so true when we start thinking about, the function of specific genes in different organisms, that the organism that you're studying really, really matters.
Do you know, by the way, when we had Esther Angert on the show, at one point we were talking about this as part of discussions of the ginormous bacterium E. pulopicium. And she said, and I brought this up, and she says, well, what's true for E. coli is true for E. coli. And then she paused and said, some of the time. And that's just perfect.
Yes, that's absolutely the perfect saying, that what's true for E. coli is true for some strains of E. coli.
Yes.
I think that's...
And your third?
And then my third takeaway is that I think we really have so many exciting discoveries that are still out there to be made about gene function, that this is something that I think the computational developments recently are really critical, really critical for meshing this together with the sort of experimental phenotypic driven screens. But that, in bacteriology, there's so many exciting discoveries and so many functions of so many genes that are just kind of out there waiting to be discovered.
Well, I have to say how much I've enjoyed this session, and not just because, as we talked, I remembered seeing one of your seminars at the West Coast Bacterial Physiology meetings.
That's right.
Probably more than once, if you went more than once.
Mm-hmm.
Yeah.
Yep.
So it is indeed a small microbial world in which we all inhabit.
Yes, it absolutely is.
And so I want to wish you and your lab and everyone else in your life the very, very best. And thank you so much for your time and your enthusiasm.
Thank you so much for having me. It has been an absolute delight to have this this fun chat about microbial dark matter and about really the small world of science. And yeah, thank you again for inviting me.
This has been Matters Microbial, a podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, with, as always, awe-inspiring links, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Julia Willett can be found in the Department of Microbiology and Immunology at the University of Minnesota in snowy Minneapolis, Minnesota. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our Quality Quorum today. See you next time on Matters Microbial.